Hey CW Bio, Mr. Kennedy here. Today, I'm going to do a whole bunch of practice problems with you to help you understand how the Punnet squares related to complex human hereditary patterns work. We're going to look at incomplete and codominance. We're going to look at multiple allele inheritance and sex linkage to be specific. To start things off, we're going to get into incomplete and codominance. So grab yourself a sheet of paper, grab yourself something to write with, get yourself a comfy chair and something to drink, because here we go. On your paper, first thing we're going to do is we're going to write incomplete dominance. Okay. In incomplete dominance, just like codominance, you have a situation where, well, you get a third phenotype in your offspring that you may not necessarily have expected. In incomplete dominance, the reason you get that third phenotype is because, well, you can inherit one working gene from one parent and one gene that's not working very well from the other. So rather than get like true distinct separate phenotypes, you get something that in the end kind of looks like a blend. In the previous video on this that I did for you, we were talking about red and white snapdragons, and there I told you that if you crossed a red flowering snapdragon with a white flowering snapdragon, you would get pink flowering snapdragons. And it's not because somehow the white gene is like intermingled with the red gene. It's just that the white gene doesn't work and the red gene does. And if you're a heterozygote and you get one working gene and one that doesn't work so well, then you don't really produce enough red pigment to be red. You end up looking pink, okay? So I'm going to write incomplete dominance like this and say that one trait cannot hide the other. And in parentheses, I'm going to write the word blend, okay? Because the offspring that you get are going to look like a blend between the two parents. Then I'm going to do codominance. Codominance. Okay, in codominance, basically both genes are expressed equally. Both genes expressed. So in the case of our snapdragons, instead of getting pink, you're going to get like, red and white stripes. So I'll just write stripes. Okay. All right. So red flower crossed with white flower gives you like red and white striped flower or stripes in your flower. Okay. So let's do a couple crosses and see how this works. I don't want to just like rehash something that you've seen in your book. So let's, I don't know, let's talk about chickens. Chickens show incomplete and or codominance for feather color. So let's write this out. Chickens show incomplete / codominance for feather color. All right. So if they show incomplete or codominance for feather color, what we're going to do is we're going to cross a pure breeding black feathered chicken with a pure breeding white colored chicken and see what we get. Okay. So if you're pure breeding black, all right, that means that the first parent should be big B for black. And since we're talking about incomplete and or codominance, then the other parent that is pure breeding white, well, they aren't little b. They would be big B prime, big B prime. So in essence, they both look like they're homozygous dominant, but they're really not, okay? It's just that, the black gene is working, producing the protein that we need, and the white gene is not, so it's got that little prime indicator, so it's not producing the black pigment, okay? So we've got the black chicken, big B, pure breeding black, crossed with a white chicken, big B prime. We are going to use a Punnet square to do this cross, just like we learned in our past notes. So we take the parents, they go through meiosis, right? And we have the first parent with big B on the side, the second parent with big B prime across the top, okay? Then we fill in our boxes like so. They should all get a big B prime, just like you see there. Okay? What's the genotype and phenotype? Well, that should be pretty easy. Genotype, you guys should be good at this by now. We got 4 out of 4 boxes that have big B prime, so we're just going to go straight to 100% big B prime. That's your genotype. Phenotype, again, you should be good at this by now. So what color are those chickens? Well, that really depends. Notice up here I said incomplete and codominance. Depending on which one you're talking about, well, the color of the chickens will change. So here we are crossing basically and I'll put it underneath to help you, a black feathered chicken with a white feathered chicken, okay? And they're pure breeding for those colors. Over here, we get what is only describable as a heterozygote, right? Big B prime. But what color is that? Well, if it was incomplete dominant, then the color would be 100% big B prime, and our chickens would probably be like, gray. Okay? If it was codominance, okay, we'd still be 100% big B prime, but maybe your chickens look like, I don't know, checkered. They've got both black and white feathers, so we'll just put checkered. All right? If they're big B prime, okay? And it's codominant, because both of the genes are going to be expressed. Make sense so far? I hope so. So we're going to do one more cross. What would happen if we took 2 chickens that were like this from the F1 and we crossed them? So in words, basically I'm crossing 2 heterozygous chickens. Or if I wanted to focus on incomplete and codominance, I could say I'm going to cross 2 chickens that are gray if it was incomplete, or I could say 2 checkered chickens if I was talking about codominance. At the end of the day, it's big B prime crossed with big B prime. Okay? We do the same Punnet square that we've done in the past. The parents undergo meiosis, produce their sex cells. Okay? And we get one parent on each side like so. Okay? Fill in the boxes. First box gets big B, then a big B prime, big B prime, and then 2 B primes. Okay? Genotype and phenotype. All right. So genotype. I've got 1 big B. I've got 2 big B prime, and I've got 1 B prime. Okay? So that's my genotype. Phenotype. Phenotype is going to be very similar to your genotype. I'm going to have 1 where I just put big B because that's the color. And I'm going to have 2, which are going to be big B prime. And then I'm going to have 1 just straight B prime. Now remember, if it's incomplete dominant, okay, the phenotype will look different than if it is codominance. So I'll put both underneath. If it's incomplete dominant, okay, then your big B is black, your B prime is white, and then the 2 in the middle, the heterozygous ones, if it's incomplete dominance, right, they should be gray. Okay, and codominance, still black for the B, still white for the B prime, and the 2 in the middle would be checkered. Okay, so that's how you do incomplete and codominance. The big deal here is just that, the letters look a little bit different, and the phenotypes change a little bit, but nothing I'm sure you can't handle. All right, practice it to make sure you get it locked in before test day. Next thing we're going to look at is sex-linked inheritance. Okay, so in sex-linked inheritance, we have to revisit the idea that in order to determine an individual's gender, sex-linked, right, heredity, In order to determine an individual's gender, we have to go by the chromosomal standard of XX equals female and XY equals male. Okay? So, at the outset, it's important for you to remember, who determines gender and how these things are passed on from one generation to the next. So if we just do a very simple, plain Jane cross like this between a male and a female, just looking at the sex chromosomes, we do a Punnett square, right? Simple monohybrid cross. We can put the female on the side, the male across the top, fill in our boxes. Okay, and then we can go genotype and phenotype from here so that you have an idea of, the ratios. So genotype, we've got 2 boxes, 2 of the 4, which are XX, and 2 of the 4, which are XY. Okay, this reduces down to a 1XX to 1XY, or in percentages, 50-50 split between male and female. Okay, so that's your genotype. And as far as phenotype is concerned, well, it's the same ratio. The only thing that would change is we would probably write the word female here and male over there because that's what you see. Okay. The last couple things that I want you to get from this Punnett square is just a couple of reminders that say it is the male that determines the gender of the offspring. And when you have those offspring, remember what we learned in that video. If it's a 50-50 chance of having a boy or a girl, what that means is every time this mating event occurs, it's always a 50-50 chance of having a boy or a girl. So there's no guarantee that your first child will be female. There's no guarantee that your first child will be male. And regardless of what gender your first child is, there's no guarantee that you'll get the opposite gender the very next time you have a child. This is just like flipping a coin. So every time you flip a coin, there's a 50-50 chance of getting heads or tails, right? Realistically, if you had 100 kids, well, about 50 of them should be boys and 50 of them should be girls. But who's going to have 100 kids? Yeah. So that is how, gender is passed on from one generation to the next. Let's look at a trait that is specifically sex-linked. And include the gender issue. So we're gonna study color blindness here. Color blindness. Okay. Color blindness is a sex-linked trait. Okay. To be sex-linked really means that you're passed from mother to son. Typically, girls carry the sex-linked trait but don't express it. I'll show you a kind of an exception to the rule where a girl can actually express the sex-linked trait, but that's not something that is terribly common. When you think about color blindness, it's not horribly common, so we're going to assume for the sake of argument that big N is dominant to little n here, and that big N stands for normal vision, which means I can see color, right? If you're little n, then you are either red, orange, or blue-green colorblind, okay? All right, so if I want to study how this is passed from one generation to the next, I'll do a practice problem. We'll do something like this. Since I told you it's passed from mother to son, and I told you it's fairly rare, let's assume we're going to take a normal vision male, right? And we're going to cross him, crossed with a female carrier. All right, that's about all we need to say, and then we can do genotype and phenotype, right? So normal vision male crossed with a female carrier. And it doesn't seem like I've given you a lot of information here to set this problem up, but I've actually given you all you need. Let's break this down. First things first, we know it's sex-linked because we're talking about male and female. So we got to put an XY for the male, and we got to put an XX for the female, okay? Second thing is the vision gene. So your sex chromosomes can occasionally carry a few traits or a few genes, and color blindness is an example of one. Now you'll notice I didn't tell you if the male was homozygous, for normal vision or heterozygous. If you have normal vision, I mean, technically, as long as you inherit that one dominant gene, you should have normal vision. But I didn't say anything about the male being heterozygous or homozygous for a reason, and that's because the male only has one X chromosome to carry this gene on. So I don't have to tell you if he's homozygous or heterozygous. If he has normal vision, the only thing he can be is that. So I put as like a superscript a capital N for normal vision. The female, you're told she is a carrier, and the word carrier is code for heterozygous. Okay, heterozygous. So that means she's going to have one big N, one little n, just like that. Now, we're going to do a Punnett square, okay? And we're going to see how the kids come out, all right? So we'll draw the Punnett square, put the male over here, the female across the top, and we'll fill the boxes in. All right, so genotype and phenotype now. So geno, we've got 1, big X, big X, big N, big N. We've got 1, big X, or just X, big N and X little n. We've got 1 X with a big N and a Y, and we've got 1 X with a little n and a Y. Okay, so that's your genotype. It's a 1 to 1 to 1 to 1 ratio. You got a 25% chance of any one of those things happening. Okay, so each of these is 25%. Now, phenotype. What do these individuals look like? If we ignore gender, okay, we can just look at the idea that this one has a big N, that one has a big N, and that one has a big N. So you're going to have 3 big N's, okay, and you're going to have 1 little n. So you got a 75% chance of having a child with normal vision, a 25% chance of having a child that is colorblind, okay? If we break it down by gender, then you have a 0 chance of having a girl that is colorblind in this scenario, and a 50% chance of having a boy that is colorblind in this scenario. And you have to be careful with your genetics questions on tests, because sometimes it will say, what proportion of the offspring are colorblind? The correct answer for that would be 25%. If it said what proportion of the boys are colorblind, then the correct answer would be 50%, because now we're just talking about boys versus the previous one, which was talking about all the offspring collectively. Okay, so you got to read questions very, very carefully. So this example also illustrates how color blindness can be passed from mother to son. So the mother up here, she carries it, but she doesn't show color blindness because she has a healthy normal vision gene to hide the colorblind gene that she's also carrying. So she's not going to be colorblind, but look what she did. She passed on her X chromosome with the colorblind gene to her son, and that is what makes this guy colorblind. You see, when dad determines the gender of the offspring, in order to make a boy, dad has to contribute the Y chromosome. The Y chromosome doesn't carry any of these traits. All it carries is a factor that determines maleness, right? So mom passes these sex-linked traits from mother to son. Now, a while ago, I said there is the potential for a girl to show a sex-linked trait. How can that happen? Well, if we stay with this scenario, where we're talking about color blindness, there is a possibility where you can have a girl be colorblind. It works like this. If you take a colorblind man, all right, so that would be X little n Y, a colorblind man, and that man has children with a woman who is a carrier, okay? It's possible, I mean, women marry colorblind men all the time. Okay, so this could happen. If the woman is a carrier for color blindness, let's look at that Punnett square and how it plays out. Okay, so the male we'll just put right there. The female we'll put over the top. Okay, and we fill our boxes in. So we've got a girl here that is a carrier. We've got a boy here with normal vision. And then, uh-oh, look at this. There's a colorblind girl, and there's a colorblind boy. Okay, so again, it's really important. We talked in that video on human hereditary disorders, how important it is for you to know your family history. So, you know kind of like what genetic diseases or disorders might be in your family. Well, here's another great case in point. If a woman's father was colorblind, there's a very high likelihood that she is a carrier for colorblindness. If she then goes and has children with a colorblind male, then as you look at this, there is a 50% chance that she's going to pass that colorblind gene on to any of her children. So your genotype, okay, would be one girl like this, one girl like that, and one boy like this, and one boy like that. That's your genotype. Your phenotype, well, if we ignore gender here and we just went with color blindness, you can see that basically for every individual that has normal vision, one big N, you're going to have one little n, okay, colorblind, all right? There are all kinds of other examples we could use here. We could use male pattern baldness. We could use royal hemophilia. I mean, the list goes on and on and on, but I think you get the idea. So this is how you do a cross for color blindness. All right. One last scenario that I want to share with you, and it involves multiple allele inheritance and our ABO blood type system. Okay, blood types, ABO blood types. So basically what I'm going to tell you is you kind of got to memorize what it means to be heterozygous and homozygous for these different blood types. Okay, so we have 4 basic blood types, A, B, AB, and O. To be homozygous for a blood type like type A, your genotype looks like this. Right? Blood type B would look like this. AB would look like this. And O would look like this. Okay? To be heterozygous, you would look like this. If you had type A, if you had type B, you would look like this. And then there is no such thing as heterozygous AB blood or heterozygous type O. They just don't exist. Okay? So these are the genotypes for the different blood types. All you have to do in order to do a cross using blood type is know this table and be able to crank out a Punnett square. Here's one last example in this video for your notes. So I'll just say I'm going to cross a heterozygous type A person with a type O blood typed person. Okay, so I set the parents up. Here's my heterozygous type A. Here's my type O. So IA, IO, cross with IO, IO. So it's off to work I go. Oh, okay. Yeah, that's something different. All right. So anyways, so then I do this Punnett square. I'll move this up because I bet you can't see this. Right? Yep. All right. So I'll do my Punnett square. Parents on the sides and top, just like we always do. All right. And we fill them in. IA, IO, IA, IO, and then IO, 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 IO. All right, genotypes and phenotypes, real simple. Matching game, okay? So I've basically got one, well, here, we'll go 1, IA, IO, to every 1, IO, IO. There's my genotype. That's a little small, but if you squint, you'll be able to see it. All right? And for phenotype, okay, we have 1 heterozygous type A. So 1 type A, and we can put hetero underneath it. So we know it's heterozygous to 1 type O. Okay? that's how you do those Punnett squares. It's super easy, super simple. So don't make it harder than it has to be. The key to doing these is you have to like deal with that table. Okay. Outside of that, it's easy peasy lemon squeezy. So you might be asked on a test to do what I just did, cross a heterozygous type A with a type O, or you might be asked a question along the lines of like, hey, you've got a dad who has like type A blood, a mom that has type O blood, and then here's these 4 kids, and here's their blood types. Which of those kids with those 4 blood types belong to those parents? Right? So you'd have to do a Punnett square to know that those parents, if the dad was heterozygous type A and mom was type O, you'd have to do the Punnett square to know that they could only have kids that had either type A or type O blood. So if there was a kid sitting there with type AB blood, then you got to go, whoa, that kid doesn't belong to those parents. All right, ladies and gents, that's how you do Punnett squares using complex human hereditary scenarios. I hope you've learned lots. Practice this, practice till you get it down. I'm Mr. Kennedy, and I'll see you next time.